Hello everyone, my name is Asisipho and welcome to my YouTube channel. In today's video, we'll be answering this female reproductive system question from a previous Life Sciences paper 1 question paper. The diagram below represents the female reproductive system. 221, give the letter and name of the part where. So for 2 marks, guys, they are asking us for both the letter and name. Remember, it's very important that we read and understand these questions, guys. Sometimes they ask for just the letter, sometimes they ask for just the name, and sometimes they ask for both. So the letter and name of the part where fertilization takes place. Fertilization will take place in the fallopian tube, which is B in our diagram. So the letter is B, and the name, it's the fallopian tube. You then get a mark for the letter in the name. This is you. So mark for the letter, mark for the name. Letter and name of the part where the embryo develops, where the embryo develops. That will be in the endometrium, which is part of the uterus. So C. So C is the letter and the name, uterus. So then mark for the name, mark for the letter. State 2 functions of part D. So they are not asking us to label part D, they are asking us to just give 2 functions, and that's what we need to do. So even if you do mention that it is the vagina, you're not going to get a mark for it, so there's no need for you to mention. So the 2 functions, we can talk about the first one being the birth canal. So we know that the vagina is a birth canal, birth canal. Also, this is where the penis will be placed during sexual intercourse or during copulation. So we can say it receives the penis during copulation. This is also the place or the structure where semen will be released into during ejaculation. So we can also talk about that. Remember, they asked for 2, guys, so you just need to mention 2. So place where semen is released during ejaculation. 2, 2, 3. We'll just mark the first 2, guys. First 2. Okay. So 2, 2, 3. Name and describe the type of gametogenesis that takes place in part A. 5 marks. Oh, they love this question. We need to name and describe. So we're going to name first, and the type of gametogenesis taking place in the ovaries is oogenesis. Oogenesis, that's the first mark, and I think that will be the compulsory mark. So that means the process itself, when you describe the process itself, it will be marked out of 4. Okay, so let me write the process here. I always talk about the importance of understanding oogenesis, how to describe oogenesis and spermatogenesis. So as mentioned already, oogenesis is the type of gametogenesis that will take place in the ovaries. That's the first mark. Now, when you describe the process, you obviously need to talk about the diploid cells. These diploid cells are first going to undergo mitosis. So the diploid cells in the ovary, this is an O, in the ovary will undergo mitosis first, and they will undergo mitosis to form numerous follicles. Then under the influence of FSH, this will be during puberty. So during puberty, under the influence of FSH, under the influence of FSH, one cell from those numerous follicles, one cell inside the follicle will enlarge and will then undergo meiosis. So it's mitosis first, then meiosis. Will then undergo meiosis to form a haploid ovum. You can even talk about undergoing meiosis to form cells, but out of those 4 cells, only one matures to become a haploid ovum. Actually, I think that's how it is in the marking guideline. How will this be marked? Let me change my color here. So the first mark will be the compulsory mark. So 1 mark for naming the gametogenesis process. Then the second mark will be talking about diploid cells undergoing mitosis, undergoing mitosis to form numerous follicles during puberty under the influence of FSH. One cell is going to divide to form mitosis, one cell inside the follicle, will enlarge and undergo meiosis to form a haploid ovum. So I know there's 5 ticks for the describing part, but the teacher will mark any 4. All right. We need to draw a labeled, we are working here. Draw a labeled diagram to show the structure of a haploid cell that will fuse with the haploid cell produced in part A. Okay, let's see. Part A is the ovary and it will produce the ovum, right? So they are asking us to draw the label diagram of the sperm cell, because the sperm cell is the one that is going to fuse with the ovum. Do I even have space, guys? Oh. So remember, the sperm contains the head, and it's not going to come out perfectly, guys, for my diagram because I'm not even using a normal pen. So let me try my best. So that will be the head, right? And then you will then have your middle piece with mitochondria. Then you have your tail. Make sure that yours is nicely proportioned, guys. Mine is coming out horribly. Oh, goodness. My God. Okay. Guys, please make sure that you follow all drawing and labeling rules, please. Please make sure that you follow all drawing and labeling rules. This will obviously need a caption. That is the label diagram of the sperm cell. Then we need to label all the structures. So from the head, so this will be the head, and this will be the middle piece, and this will be the tail. Okay, then in the head we have the acrosome, then we have the nucleus. I am very unhappy with my diagram, but hey. Then you will have the mitochondria. It's mitochondria. There's an A at the end, guys, and that's obviously the tail. Okay, very unhappy with my diagram. How many marks are located here? 5 marks. Okay, so it's 5 marks. This is how it's going to be marked. 1 mark for the caption. So the caption is very important. This is the diagram of the sperm cell. Then the correct diagram itself. So if your diagram is correct, well proportioned, we can see the head, the middle piece with mitochondria and the tail, you then get a mark. I think the code will be CDL. CDL. C for the caption, D for the drawing, then L, 3 correct labels, 1, 2, 3. So if this is correct and this is correct and this is correct, you will then be marked for those labels. Then that's out of 5. Shoo, I hope that makes sense. Then 2.4, this video is becoming long now. Oh my God. The 2.4, the table below indicates the growth of a human fetus 6 months after fertilization has taken place. Between which 2 consecutive months did fetal growth double. So that is 7.5. That is month 3 and 4. 3 and 4. There you go. What is the percentage increase in fetal length between the third and sixth month? Between the third and the sixth month after fertilization, show all your calculations. Percentage increase of the fetal length. So let's see, what do we have at month 6? So that is 30, the length is 30. Then month 3 is 7.5, then month 3 is 7.5. Then we multiply that by 100 and we get our answer. Okay, 300%. Okay, last one. Draw a line graph to represent the data in the table. Is it data or data? Anyways, line graph. We need to follow all drawing rules for the line graph. The caption, the scale, the labeling, everything. Mine will not come out perfectly, but I'm going to try. Let's draw the line graph. Let's see what we have here. Line. Oh man, let me use a different color because I'm going to use red to mark. So the caption, a line graph showing, remember guys, this is how you need to write your caption for your graphs. Hey, a line graph showing the length. You're basically telling us what your graph is about. So showing the length of the fetus in centimeters after 6 months. You can even talk about after 6 months from fertilization. This was an S. Okay, then there is our graph. I'm not using a ruler, so my graph is not going to be perfect, but obviously you will use your ruler. Okay, so from the memo, what we're having is just months over here, right? Because that's what we have in our table. Let's see. Yeah, so months after fertilization and length of the fetus in centimeters. I'm going to rectify something. Length of the fetus, and the length is in centimeters. Now, this will technically be incorrect. If I leave my x-axis label as months, that is technically incorrect. But I can see on the memo, they just have it as months. So you need to basically tell us that this is actually time that is measured in months. So now it's correct. We're here to learn, guys. I want you to get full marks in your test and exams. So time is measured in seconds, minutes, hours, days, months, years, and all of that. So we can't just leave it at months. It's essentially time that is measured in months. Okay? And what we're going to have on our x-axis, whether you indicate x and y, guys, it doesn't really matter, but you can indicate x and y, then you have your 0 there. It's up to you whether you have the 0 or not. So in terms of months, we have month 1 to 6. You use a ruler. 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Then length, let's look at our data. For length, 0 0.5, 4, 7.5, 15, 25, 30. We can use intervals of, it's tricky because now we're having very low numbers and very high numbers. 2, 4, 6, 3, 6, 9, 12, 2, 4, 6. Will we get to 30? Okay, let's see. 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30. Guys, like I'm saying, I'm not using a ruler. You need to use a ruler to make sure that the spaces in between your scale lines are equal. Please make sure. Then I'm going to label 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30. So I would also make sure that the spaces are big enough. Like in my graph, the spaces are not big enough, which will obviously make it a bit tricky for plotting. Make sure spaces are big enough, especially because we now have that 0.5 and 7.5. Okay, let me just actually take a picture of the table so that I don't keep on scrolling up, because that's just making the video to be way too long. Let's see. So month one, 0.5. So one will be over here halfway, then 0.5 will be somewhere here. Guys, you just need to learn the skills of drawing, hey, because my graph is now turning out to be incorrect and I hate it. Yeah, because but I don't have a ruler, guys. Then 2 is at 4, so this is 2 4, so 2 is at 4. And then 3 7.5, there is 3, there's a 6 and a 8, there's a 7 7.5 will be somewhere there. Okay, then 4 is at 15 4, 15 is between 14 and 16, so somewhere there. Then 5 is 25, 25 will be between 24 and 26. So you're nicely scaling it, guys, and plotting correctly using your ruler, please. 6 is at 30. Then you join your dots. Remember to not use a ruler when joining the dots. Do not use a ruler. Okay, yeah, there's your graph. Let me show you how this will be marked. TCLSP. T is the type of graph. Line graph, 1 mark. Caption, a line graph showing the length. That's your heading. Labels, you've labeled your x-axis and your y-axis with the units. Scale, your scale increments, your scale lines and everything for both the x-axis, your intervals, equal increments, everything, and you have your scale lines and your y-axis. You get a mark. Plotting, all the points plotted correctly. That is 2 marks. Okay, we're done.